I finished my bachelor in sustainable biotechnology in Denmark in 2016, whereafter I thought I want to go see the world. So I went to Australia and did a master in molecular biology at UQ. And then I was lucky enough to get a position as junior bioprocess scientist in the research and development and upscaling part of a company called Molina Bio. In 2020, Corona hit, and I thought it's about time to get back home. And I was lucky enough that Klaus had a position as a research assistant at DTU, and I was selected to be filling out that role. So I started as a research assistant, and that turned into becoming a PhD student later in 2020. So a little bit of the project background. In the future, we're going to be 10 billion people, and that's a whole lot of people. And we need to provide food for everyone. In order to do this, we also need to provide more food than we currently are making. But if we want to do it, we should do it without deforestation. We don't need more cropland. We don't need more grazing areas. We need to reduce the amount of area that we're already using for doing agriculture for sustainable and environmental reasons. And we are also looking into removing the animal part of the food that we're producing, so that we don't need to think about crops for feed. We don't need to think about the fertilizers and biocides that comes with that, the water consumptions and the resources used in transportation and slaughterhouses. So in general, these are things we don't want, but what we do want is to keep it healthy and safe and nutritious. And the way that we can keep it healthy and safe without using new land and without introducing animal products is by looking at side streams. So side streams are these large byproducts from the industries. Some of the side streams we're looking into are rapeseed pulp, brewer spent grain, apple pulp, and sugar beet pulp. So it's all side products from some kind of industry. And right now, these side streams are being used for energy production, so through combustion, but they're also being used to produce methane, which can be used both as a fuel, but also for energy production. And it's also being used for feedstock, so that they're producing animal products from that. But there's a lot of problems with using it for these things. One thing is that the side streams spoil very easily. So you have to do something to it. So you have to either dry it or to press it or to freeze it in order for it to be able to conserve for a longer time. So you can use it for these feeds or other things. And there is also a problem with using it for feedstock, because the conversion rate from feed to animal protein is very low. So we can feed them a lot of plant protein, and then the production of protein would be low compared to what we would give them. So what we're looking into, I'm not sure if you can see my mouse, but I'll try to put a pointer here. So we're looking into using lactic acid bacteria together with fungal mycelium and do different fermentations of these things, of these side streams, so we can produce protein-rich mycelium that we then can use for different alternatives to meat. And one of the things that we're doing, using for doing this is the DTU cell bank. And this cell bank is actually one that Klaus and his team has established all over the years. And I have been a little bit part of that as well. So we have more than 1,500 strains isolated from different sources in Denmark. This includes strains of yeast, of bacteria, and of different fungi. And many of the samples have been isolated either from plants, from fermented foods, or insect microbiomes. And the reason why we're doing these different niches is that we are looking to find very specialized strains. So if you're looking into growing something in dairy, wanting to make some dairy products, then we're looking at using bacteria we have isolated from dairy. Or if you want to look into degrading some cellulose or something similar, you would look into yeast or bacteria or fungi that we have separated or isolated from cellulosic material. So we also have this versatile uses that I'm talking about, that they're highly specific trains or highly specified trains, but we have such a broad range of cells of different strains that we can find strains that could work for any type of media. And we also have a lot of genomic data from a lot of the different species that we have isolated, and we have found out the majority of the strains that we have are also QPS, which is qualified presumption of safety. So they're safe to use in food as long as they don't contain any anti-resistance genes to antibiotics. And the way that this cell bank has been created is that we go out in nature, we take some samples from soil, from fermented foods, from different things, and we plate it on agar plates. Then we select different colonies, and we go and identify what we have found with MALDI-TOF to make sure that it's something safe, and to make sure that we know what we have in our cell bank before we start doing all the genomic data. And then we isolate DNA, go with get out the DNA extraction to get the genomic data, and then we can use it for very different types of fermentations. So for what I have done previously, I have been looking at 35 different strains of brown rot fungi and white rot fungi, which I have tried to grow together with different lactic acid bacteria. And to screen for which one might be good with lactic acid bacteria, I grew them on plates with lactic acid, low pH, and the ones that had bad growth, I disregarded. And the ones that had good growth, I selected for testing together with lactic acid bacteria. Then I would test them with and without lactic acid bacteria and compare how they grew together. And here are some results for this. So I took these pictures a few days after the experiment was done. And these round circles are where the mycelium grew to during my experiment. So the last one is for 8 days of growth. And this one is the control. So it just grows without any bacteria, and it had half a plate full at 8 days. Then I grew it together with one of the bacteria, and it had almost full plate growth at 8 days. And at another sample, I also grew it with another type of lactic acid bacteria, where it had very targeted spread towards the bacteria instead of trying to fill out the plate. But the mycelia, as you can see here, is also much thinner. So even though they're both lactic acid bacteria, they have these different influences on how the mycelia comes out in the end. And then a different view, I had this strain 330 that grew very well without lactic acid bacteria. When I then grew it with lactic acid, with the bacteria, it had very inhibited growth already from the first few days. And on day 8, it would barely spread any further. And when I grew it with another lactic acid bacteria, it showed some inhibition. It grew tall instead of wide, but it could still survive. So there are these differences. And I'm a little sorry if you can hear my son in the background. My husband has him under control, so don't worry. But he's a little loud right now. All right, liquid co-fermentations. So after I looked at these fungal strains, which showed really good growth together with the lactic acid bacteria, I then tried to grow them in a liquid culture together with the bacteria to see if they would change from what we saw on the plate. And I found some really interesting results. So for this strain, it grew pretty well without any bacteria. And then I tried to acidify the media it grew in, and it's changed the color to a darker mycelium, and the growth also increased. But when I grew it with one of the lactic acid bacteria, instead of having this pellet-like growth when I have homogenized the mycelium from the beginning, it would merge all these pellets into one big mycelial lump, which I found very interesting because that could make it easier to harvest in the end. And I saw the same for one of the other lactic acid strains, but this one didn't promote as good growth as the other one. So even though it promoted this difference in morphology, there are still differences between just 2 strains of lactic acid bacteria. So here I drew another type of fungi, and it really didn't grow very well just on its own. it had a lot more growth when it was growing in the acidified media, and again it turned dark, which can happen when you grow them in acidic conditions. But when I grew it together with the lactic acid bacteria, it had increased growth, but it didn't turn as dark as we saw when it was just acidified. And then down here is the most interesting, I would say. We have another strain of fungi that grows alone, and in acidified media, it again changed the color. But when it grows together with one of our lactic acid bacteria, it turns this orange-red color instead. And it only happened with one of the bacteria, where with the other one, it had some increased growth, but it was not as good. And it's still turning some brown. So we found out later that this might actually be because the fungal strain is producing a type of acid, a fungal acid, that also works as a pigment. So it's very interesting that just growing it together with a bacteria, it can induce these changes. And to try and figure out what was happening, I tried to separate in agar. So we have these agar plates that has been separated with a wall of glass in the middle, and you pour agar on each side, and then you put a fungal strain on one side and a bacterial strain on the other side. The top one is without fungal strain, just to see how the without bacteria strain, just to see how the fungi grows alone. And then here it's with one type of lactic acid bacteria, and here with another type. And on day 4, we already see there's an increased growth of the fungal strain, even though the lactic acid bacteria does not get in contact with the agar that the fungal strain is growing on. And on day 8, it has taken over the plate completely, where the control and the lactic acid bacteria has not. So we see this change in this induced growth, which cannot be explained by it just being acidified. So we were looking into maybe this bacterial strain is producing some volatiles that could induce this growth. And here are some of our results. These are early results, so we haven't finished on these yet. But what we're looking for is some kind of volatile compound that we find in only when this strain is going together with the bacterial strain and not in any of the other cases. So we're going to publish this pretty soon, but we're still trying to find out exactly which one could induce this growth behavior in the fungal strain. And other current work that I'm working on right now. So this is some work that one of my students is working on. I'm helping her a little bit, but we're looking into industrial size strains, as explained before. And we have found some of these interesting fungal strains from my previous experiments that we want to know, can they grow on any of these size strains? And if we grow some of the lactic acid bacteria on it, can we find 2 that are really good on one of the side strains, and can they synergize when they grow together as well? So we want to combine all of the knowledge before into one project where we hope to grow a different kind of mycelia together with lactic acid bacteria to induce the growth and to get more protein out. And we also, for another work, I'm also looking into producing oyster mushroom mycelium from sugar beet leftovers. And this is a little bit different from what I've been looking at before, because oyster mushroom does not like acidic media. So when we're pre-fermenting with lactic acid bacteria, we're not doing it in order to increase the mycelium. We're actually doing it as there are 2 reasons for that. One way is that we hope to be able to use sugar beet pulp fresh. Right now we're using a dried sugar beet pulp, which it takes a lot of energy and a lot of time and problems with transport and such, because you need this sugar beet pulp to be treated right after it has been the sugar has been extracted from it. And instead of having to dry it and then rehydrate it when we need to use it, we hope to get the fresh pulp and then ferment it with lactic acid bacteria, so it's conserved. And then when we need to use the mycelium, we just increase the pH and then ferment. That is the idea. We are also hoping that the lactic acid bacteria can remove some of the off flavors that we see are in sugar beet pulp. So by pre-fermenting, we hope that there is some sandiness and some other off flavors that we hope that these will not be present in the mycelium powder in the end. So after the fermentation of the mycelium, with the mycelium, we can either dry the mycelium and get a mycelium powder, which can be added as an ingredient to a meat substitute, maybe like a flavor ingredient or for increasing the protein content, or we can actually use it directly as a meat substitute. We found we did a prototype not long ago. Here is the picture of the prototype. And this is mycelium from the fermentation that has just been hand pressed after the fermentation. And the texture of this is like somewhat spongy, a little bit like a meatball. And then it was fried on the outside, so it becomes a little bit crispy, just like when you're frying meatballs. There are still a bit of problems with the off flavors, so we're trying to see if post-fermentation might be able to remove these, or if we have to solve it through some other ways. But we're working to solve this issue. And then the last thing that I want to present is, so we have been growing oyster mushroom in sugar beet pulp, but there is a lot of other side streams out there that might be better for growing oyster mushroom. And we're looking into brewer's span grain, sugar beet pulp again, to have something to compare with, apple pomace, rapeseed cake, and then a control on malt extract. So this is only on 10%. We had different concentrations, but this is just to give an overview. And what we see is that it looks like the brewer's span grain is actually better than the sugar beet pulp. And it could just be because it's higher concentration here, but we saw the same with the lower concentrations, that brewer's span grain might be a better media for it. So brewer's span grain is a little bit complicated because different breweries have different brewing processes. So you need to get the span grain from the same brewery every time in order to get the same results. And it could also vary between countries. If someone was to do the same as we have done, maybe it would look different. But at least this one that we get from Carlsberg is very good for our oyster mushroom. So we wanted to try in liquid cultures as well to see if we have the same effect in the brewer's span grain as we got on the plates. And here we have the control, and we have 3 different concentrations. So we have the highest concentration here, a medium concentration, and a low concentration. And this is after 4 days of growth, and we see no mycelium in the control. But with the mycelium at the highest concentration, it looks like it's a bit inhibited. And at the medium concentration, it's growing pretty well. But at the lowest concentration, it's growing really, really well. So after 8 days, which we have done afterwards, we see there's an increase also in, sorry, I just got some pop-up. We saw an increase in the mycelium in the highest concentration as well. So it seems that the amount of sugars that might be available might be inhibiting a bit the mycelium. So it has to get used to this substrate first before it starts growing. But that could be maybe countered by adding more mycelium from the beginning. But it could also just be, that some of the others are better to go with. So in conclusion, interactions between edible fungal mycelium and lactic acid bacteria can be beneficial for preserving the substrate, and in some cases can promote the growth of the mycelium and even induce morphological changes. Cross-kingdom fermentations between these 2 types have been previously untapped into, and it's looking very promising for trying to convert side streams into protein-rich foods. Thank you for your attention. Do you have any questions?
Thank you, Sanne. So we have time for some questions.
Yeah.
I have a question actually.
Yeah.
Yeah. Have you found any volatile that can be relevant on the growth of the fungi? Yet, or?
So I haven't tested it out yet, but right now it looks like the acetate could be the reason why it's changing this way. But I have to test out the fungal strain and then just drops of acetate on the other side to see if there's any changes. I haven't done that yet.
Nice. That's so cool. Nice.
Yeah.
Some other question? We are.
Hello, thank you for your presentation. I have a question. What kind of flavors do you expect to remove from the beet pulp with the lactic acid fermentation?
Yeah, so we have something called hexanal, which gives a like sandy green kind of off flavor, and we are hoping that by using the lactic acid bacteria, it could remove some of them. I haven't looked into pre and post fermentation of every volatile and every flavor that is out there, but there are some of them which are being reduced after the bacterial fermentation. And we have done, one of our colleagues have done a testing of the taste of this mycelium before, when it's not grown with lactic acid bacteria and when it's grown with lactic acid bacteria. And she did notice that there was this reduction in the sandy taste. So hopefully if we do post-fermentation, we'll see an even further reduction.
Thank you.
And this is also actually something I think Guillermo will elucidate a little bit more on the matter of removal of anti-nutritional and off flavors.
All right. I will stop the sharing.
Thank you, Sanne. And now Guillermo is going to present his thesis project, whose research is in plant-based fermented dairy alternative applied microbiology. So thank you, Guillermo, for being here also.
Thank you a lot for inviting us, and then we can share a bit of our projects. So I'll start sharing my screen. And let me just find it. Okay. So do I have the same problem as Sanne has?
Yes.
It's not on full screen.
Yes, you do.
Okay, now, is it full
Yes.
screen now? Okay, cool.
Yeah.
So I'm Guillermo. I'm the PhD student in DTU as well as Sanne, and one of the PhD students from Klaus group. And my project is called Replanted. So basically, it's related to the detection and removal of undesirable compounds for the generation of plant-based dairy alternative by lactic acid bacteria fermentations. So I'll try to introduce myself a bit before starting with my talk. So I did my bachelor's in biotechnology, and I finished it in 2018 in Barcelona because I'm from Barcelona. Then I moved to Denmark to do my master's in food technology. So, and I started my PhD 7 months ago, approximately, in microbial biotechnology applied to food to food fermentations in Technical University of Denmark. Then I have also some work experience in different food industries and as researcher in other kind of fields different than food. But yeah. So this is the agenda for my presentation. First of all, I'm gonna introduce a little bit my project, Replanted. Then I will give you a little bit of introduction to the main off flavors and anti-nutrients that we can find in the raw materials that I'm working with. Then a bit of introduction to lactic acid bacteria in relation to the removal of unwanted compounds. Then I will just mention some of the projects that maybe you are gonna be interested in that I'm working in now. And then I'll introduce some of the lactic acid bacteria that we have lately been working on. It's basically from insects microbiomes that we have found some applications for food and biotechnological purposes. And then I'll explain a bit about the project that we are almost publishing, or at least submitting to a journal, and with some results and some conclusions. So this is the overall flow diagram of my project. So Replanted, the aim is to develop processes and cultures to produce plant-based dairy alternatives to milk products. So basically, yogurt, plant-based yogurts, plant-based cheeses, plant-based milk alternatives. So it could be kefir, or it could be other kind of milk products from other kind of countries or cultures. So basically, I'm working with 3 raw materials and that are typically grown in Denmark and also around the Nordic countries, which are yellow split peas, potato, and oats. So basically, I don't know if I can, I'll put my laser as well. So basically, this is the raw materials that I'm working now. And the idea is to remove all these off flavors, all these flavors that give green, grassy, or like more like mushroom notes in order to create these milk products, fermented products. Also, the idea is to remove some of anti-nutrients that are present in these raw materials. But at the same time, we need to produce these products similar to milk fermented products. So at the same time, we need to produce some dairy flavors as well as some texture and organoleptic properties similar to what we find in fermented milk products. So basically, this is the flow diagram of my project, kind of. So I'm studying some of the genotypes or genes that can be related to the removal of all flavors and anti-nutrients. And I try to correlate it to phenotypical characterization studies. And then we try to set up some fermentation platforms using these raw materials in combination as well, and try to analyze different key quality parameters like flavor, texture, and nutritional values in order to try to mimic as the fermented milk products. So, and we are now actually developing some new or high technology in order to perform all these goals, which is droplet-based microfluidics that I will talk a little bit later. And then we also use Fourier transform infrared spectroscopy in order to analyze some of the compounds that we are interested in. But first of all, I wanted to introduce you a little bit about the different off flavors and anti-nutrients that we can find in these raw materials that I'm working with. So as you can see here in this table, in legumes, for example, peas, we find some of anti-nutrients like phytic acid, saponins, tannins that can give health problems or also affect to the viability of different minerals that can be found in these raw materials, and we cannot absorb them after eating them. So that's a problem if we want to reach high nutritional products. Then also, for example, in potato, we have cyanide, which is like a toxic compound coming from cyanidric acid. So we try to remove all these anti-nutrients from using fermentation as the main platform. Here in this diagram, you can see some of the different categories that we can be focused on. We can be focused on technical functionality, so we're trying to improve stability and texture, and then in terms of sensory profiles, we are mostly focused on flavor and taste. And then in nutritionally wise, we try to produce some functional compounds that those lactic acid bacteria strains can secrete and produce while they are growing these raw materials, as well as eliminate some of the major anti-nutrients that decrease the nutritional value of our fermented products. So basically the main concerns about these different anti-nutrients are, for example, as an example, I'm not going to go through all of them, But for example, phytic acid forms complexes with metal ions, and as I mentioned before, it chelates with some of the minerals that you can find in potato, pea, and different legumes as well, and reduces the mineral bioavailability, which means that all these minerals that can be useful for our health and our nutrition are not bioavailable for us, and we cannot absorb them. So then they became useless if we don't do something in regards to. And also in your right, you have some of the off flavors that are mainly found in plant-based. They mainly come from lipid oxidation. So as you can see here in this nice diagram that I took from a paper, so linoleic acid is the major unsaturated fatty acid that you can find in the majority of plant-based foods, and then based on one enzyme which is called lipoxygenase, it creates different, after a multiple sequence of reactions, it creates a lot of aldehydes and furans and other kind of volatiles that can impact the overall flavor profile of our foods. And if we want to make plant-based dairy alternatives, we need to remove all of them, because otherwise we can never mimic milk fermented products as yogurt. So a bit of lactic acid bacteria. So I'm focused basically in bacteria, specifically with lactic acid bacteria, which is a group of gram-positive bacteria, non-sporulating, anaerobic. Yeah, there are different species, but the main compound that they produce is lactic acid, which makes the decrease of pH, and that's what it makes partly the sourness of the fermented milk products. So here you have some of the genus that belong to lactic acid bacteria group, but basically we are more focused on the species that are approved by EFSA. We have here our European Food Safety Authority in Europe that considers some of the species being able to be used in the food industry and some not, based on different pathogenic studies and more characterization studies. So these are some of the species or genus that we can use for our purposes. So for example, Lactococcus, Streptococcus, Leuconostox, etc. A lot of lactobacilli. And then in our group, as you probably know, there are many different applications of lactic acid bacteria, but we are basically focused on non-dairy foods. So in order to understand how different off flavors and dairy-like flavors and anti-nutrients can be removed by fermentation, we need to study some of the metabolic pathways that are involved in these performances. So basically, I'm more focused on the lactic acid bacteria that come from insects and plants. As Sanne mentioned before, depending on the purpose that you want to use them, it's better to use the ones that are isolated from similar sources as your raw materials to ferment. So yeah, basically insects, mostly bumblebees and honeybees, but also all kind of plants. So here are some of the pathways that I'm focusing on. Basically phenolic acid metabolism, which also imparts some of flavors or bitterness to the products, which is we are trying to study how to remove those by fermentation and going to the metabolic pathways insights. Then we have also flavors and different pathways that can be related to this, and also saponins, for example. But each raw material has its own unwanted compounds, so it's like a complex matrix, because what it can be useful for pea maybe it's not useful for potatoes. So it needs to be, it has to be studied properly. Then here are some of the metabolic insights of some of the pathways that are involved in the production of dairy-like flavors. For example, dairy-like flavors can come from amino acid metabolism, from these leucine, isoleucine, and valine, producing these alcohols that are found in different cheeses like Gouda, Emmental, and others. Then also we studied pyruvate metabolism that is the pathway responsible for the production of other kind of dairy-like molecules as diacetyl, acetoin, and 2,3-butanediol, which give this butter aroma. And also there's a really important compound in, for example, yogurt production, which is called acetaldehyde, which its production based on lactic acid bacteria metabolism. It can come from different sources, even from DNA, but also from some amino acids and from sugars as well. So a little bit, I wanted to show you, I don't know if I can, it's a video, but yeah, maybe I can't. So basically we use droplet-based microfluidics as a screening tool. So we have, as Sanne mentioned, we have a huge collection of bacterial strains, and we need to improve our screening methods in order to find the best candidates to use for each purpose. So basically, we use one technology, which is called droplet-based microfluidics. This was a video, but I can explain you without the video. So basically, what we create is picoliter droplets, and we try to encapsulate 1 cell per droplet. And we use this platform as like picoliter bioreactors, let's say that. And we basically create an emulsion. So we put oil from the top and down, and then we put our cells and our media, and then the emulsion starts to be created. And then we collect these droplets where the cells are encapsulated and they are compartmentalized, so they grow separately from each other. And then we incubate them, and then we have something like this, like this picture. So some of the droplets are empty, so the droplets are, you can see some cells grown in them. And then after this incubation step, we do a sorting, let's say, based on fluorescence, basically. And then we select the droplets based on the fluorescent assay that you have established. Then we select the best candidates. So this is like a high throughput technology that is useful when you want to screen for the best candidates for each purpose. Yeah, now you can see the video. So yeah, basically it's how the droplets are created. So we can create thousands of droplets per second. And then this speeds up the process of screening because you can do multiple reactions at the same time, and it only takes like few hours testing maybe 1,000 strains. So for the introduction, that's it. And then I'll start introducing some of the ongoing projects that I'm involved in. So the first one is the one I'm going to talk about today. It's basically we try to study the synergetic effect of a co-culture of two different species of lactic acid bacteria, a homofermentative and an heterofermentative lactic acid bacteria, isolated from honeybees, so from insects. And we try to evaluate how they were performing in order to create these plant-based dairy alternatives based on nutritional profiles and also on flavor profiles, using soy, pea, oats, and potato milks. This project is kind of finished as the one I'm going to present, as I mentioned, but yeah, it's also. And then the second project, it's basically the formulation, because before starting fermenting whatever we want to ferment, we need to formulate the substrate for fermentation. So we are trying to formulate it different ways. using different techniques. So this second project is basically trying to optimize the formulation of the pea, oat, and potato blends. So trying to mix these three ingredients at different proportions to achieve the highest possible nutritional and texture-wise best products. And then the third one that I just started 1 month ago, it's the trying to correlate genotype and phenotype in relation to all these characteristics that I mentioned before. So removal of off flavors and anti-nutrients. So trying to dig in genomes and try to correlate it to phenotypes in order to find, as I mentioned, the best candidates. So I'm going to talk about the first project I mentioned, which you can find it here. But this project comes from 1 year ago, actually, that we published one paper that we were trying to, we were isolating novel lactic acid bacteria in order to use them as starter cultures, and we use insects as the isolation sources, because insects aren't really explored in this field in order to isolate these lactic acid bacteria strains, and maybe we thought that we could find some interesting phenotypes, and that was the case. So we published one paper, actually. So as you can see, different projects have been, we have been working on. This paper here, it's more in the biotechnological perspective, where we found out that one species from honeybees produces a really high concentration of mannitol, which is a sugar alcohol that it can be used in the food industry as a sweetener, and also it doesn't have calories. It doesn't provide calories, so it's like a non-caloric sweetener. So for the food industry, that's relevant. And we used some side streams to produce mannitol. But in this case, for now, two of the strains that we isolated in the first paper, we are using them for producing plant-based dairy alternatives. So the main goals of this project were basically creating a genotypical platform in order to study how the combination of two strains in terms of genome or genes could be relevant when we put them to grow in the raw materials that we are working with. Then the second one was in situ production of mannitol in plant. So SPOP means soy, pea, oat, and potato. That's the abbreviation. So just for you to know. So the second goal was producing mannitol while fermenting in order to increase the sweetness of plant-based dairy alternative without adding any sugars afterwards. So you can create clean label products at the end, and you don't need to add additives after fermentation. Then the second goal was removal of flavors and anti-nutrients in all these raw materials. Then study the co-fermentation or co-growth of both lactic acid bacteria strains and how they were behaving when they were co-cultured. So if they were inhibiting each other, or they were potentiating or enhancing the growth of each other, and that stuff. Then production of dairy-like flavors, and then enhancing the nutritional values of the fermented SPOP. So this is how the pipeline or the flow diagram of this project looks like. So basically, we isolated these two strains from honeybees. Then we did a genotypical characterization, taking into consideration specific genes relevant for removing these off flavors and anti-nutrients present in soy, potato, pea, and oat. Then we ferment these raw materials individually, so separate to each other, and try to analyze the fermentation performance based on pH, based on sugars and acids produced. And then the last part was the analysis of volatiles and amino acids after fermentation. So as you can see here, what we wanted is remove all flavors like hexanal, pentanal, and 2-pentylfuran, as well as producing dairy flavors at the same time as diacetyl, acetoin, etc. So first of all, this is the genomic analysis. I'm not gonna be really detailed into it, but we took some of the genes that are related to the removal of antinutrients and of flavors, as well as the production of dairy-like flavors. So we dig in all the metabolic pathways that were related to that, and then we did like a screening, a genotypical screening, and see how these two strains could be complementary to each other in terms of genotype. So, for example, I don't know. Yeah, so for example, in red, we have phenolic acid metabolism, and you can see that Lactococcus lactis possess some of the genes that are involved in the degradation of phenolic acids, while Leuconostoc pseudomesenteroides didn't. But then Leuconostoc pseudomesenteroides had a really important system, which is the proteinase, which is basically proteinases that are in the membrane of the cell, and then they can break down plant proteins or milk proteins, like any kind of proteins, and then release some of the amino acids that can be used for other kind of lactic acid bacteria to grow. If you don't possess these proteinases, sometimes the growth is complicated because you cannot degrade these plant proteins, and then you cannot acquire or uptake all these amino acids necessary for the growth. Then we did some pH analysis and growth. Here is the pH. So basically so LL means Lactococcus lactis, LP means Leuconostoc pseudomesenteroides, and CO means the co-culture of them. So basically, when they were co-culture, the pH reached was lower at the end of the fermentation. Then we analyzed also some of the sugars and acids and alcohols produced during the fermentation. So basically, you have here P fermentation using only Leuconostoc pseudomesenteroides, P fermentation using only Lactococcus lactis, and then at the end the co-culture in P. But then, for example, we have here the potato. So basically in P, the production of mannitol, which as I mentioned before, is really good as a sweetener and doesn't provide calories at all. And also it doesn't provide teeth damage. So that's also important for when we consume high amounts of sugars. So mannitol was highly produced in the co-culture in P, but for example, it wasn't that produced when we were fermenting potato, for instance. Then we analyzed some of the volatiles that were present on each plant-based raw material. So for example, here we have soy milk and oat milk fermentations after 30 hours of fermentation. And then you can see some of the off flavors, for example, hexanal. So UNF means unfermented, then LL fermented only by Lactococcus lactis, etc. And CO co-fermented. So you can see here that, for example, hexanal or other kind of, or heptanal, they are removed after fermentation when they are co-fermented. So that was one of our goals. But at the same time, we wanted to produce some dairy-like flavors. But as you can see here, for example, we have diacetyl at the down, and when Lactococcus lactis was growing alone, then some diacetyl was produced, but in co-fermentation, non-diacetyl was produced. So we were removing off flavors, but we were not producing dairy-like flavors at the same time, which was one of our goals. But then we produced some other dairy-like flavors, as this one, 3-methyl 1-butanol, which is a flavor that it comes from leucine metabolism. So it's one of the amino acids, and it's really important in the production of some cheeses because it gives this butter or dairy-like aroma. So we did this for all these raw materials, and as you can see, for example, here in oat milk, it's more clear. Lactococcus lactis alone produce a lot of diacetyl, but then when they were co-fermented, non-diacetyl was produced. But the off flavors were completely or partially reduced. Then we have the same here of our GCMS results with pea and potato milks, which, yeah, they are not milks. We changed the name to drinks, actually. But then in potato, there was something really interesting because we saw that Lactococcus lactis was dominating at the end of the co-fermentation. So Leuconostoxylus enteroides wasn't growing that much when they were co-cultured. So that implied that the production of diacetyl was higher. So you can see here in the co-culture, the diacetyl was produced in potato. This was the only raw material where diacetyl was produced after co-fermentation. So basically what we saw is that Leuconostoxylus enteroides can reduce a lot of the off flavors, while Lactococcus lactis is not that good doing that, but it's good producing dairy-like flavors. So that was the idea, to combine these two strains if they could overlap these capabilities together when they were co-fermented. But apparently we could remove all the off flavors in all raw materials, but the production of dairy-like flavors was diacetyl specifically was only produced in potato. So that suggested that maybe we should mix some of the raw materials in order to comply all our goals. And finally, we also analyzed some of the proteins, so some of the amino acids. Here is the total composition of the amino acids, but we are more interested in this diagram. So as I mentioned before, based on the genotypical characterization, we saw that Leuconostoxylus enteroides had this proteinase in the membrane that could break down plant proteins. That we saw it in the amino acid analysis, because here you have the control, so unfermented pea, fermented by Lactococcus lactis, by Leuconostoxylus enteroides, and then the co-fermentation. And for that, for all the raw materials. So you can see clearly that when you ferment only with Leuconostoxylus enteroides, the amount of amino acids released is much higher than when you ferment with Lactococcus lactis. But then when you co-ferment, then some of these amino acids are reduced compared to Leuconostoxylus enteroides alone, which means that Lactococcus lactis was taking some of these free amino acids in order for growth and for metabolic performance. So this is something similar that happens when you produce yogurt, where you have two different species, Streptococcus thermophilus and Lactobacillus bulgaricus, where you have also one proteolytic and another one that it's not proteolytic, and then they can have like these microbial interactions that they can potentiate both growth and perform better when they are co-cultured. So these are some of the of how they look like. So for example, soy at the end, it looked like some yogurt-like, more like cottage cheese. Then pea was a little bit more maybe not that thick, and then oat was more liquid, but it could be also producing some like beverages, fermented plant-based beverages. So basically our conclusions were that co-culture speeded up the fermentation and the performance in all SPOP. Then there were some microbial interactions between both strains. Basically, Leuconostoxanthus enteroides provided the proteolytic activity, while Lactococcus lactis provided the dairy-like production of flavors. Then mannitol was produced in situ, which then we can aim for clean label products because we keep the sweetness and the sourness in our final fermented products at the same time that we don't need to add any added sugars at the end. So then we removed completely or partially some of the off flavors. And then, as I mentioned before, potato was the best for diacetyl production. And then, so what we saw is that we probably need to mix some of the raw materials in order to get the highest nutritional and sensorial fermented products at the end, because potato, for example, had other amino acids that the legumes, that the pea and oat didn't have, but then pea and oat have other kind of proteins that are more functional. So yeah, I think we saw that possibly the future will be combining these three raw materials in order to create these plant-based dairy alternatives. So until here, my presentation. So if you have any questions, or yeah, you are welcome to ask.
Thank you so much, Guillermo. Very interesting, your presentation. I don't know if there is some question. No? Well, I can start with one question.
Yes.
I see that you have a lot of data. You already see all the genes that are present in these microorganisms. Are you planning to analyze the transcriptome?
Yes, that's one of the plans, because there are some antinutrients that are described of, like the metabolic pathways are described in order to understand how they are removing those or how are they interacting with those. But yeah, there are some off flavors or antinutrients that haven't been analyzed or functionally mapped. So we are planning to test some of them in transcriptomics in order to see what genes are involved in those. Yeah, that's our next plan actually. So that's nice too that you asked.
Yeah. I don't know if someone else have a question.
Oh, Guillermo, what about information for terpenoids, terpenes, remotion of terpenes? I don't know. See, you
Yes.
have information.
We haven't looked at terpenes. I don't know the metabolic pathways actually. I don't know that much about terpenes, but yeah, like all suggestions are welcome always, because yeah, there's a lot of molecules and a lot of different things that we can focus on. So it's a huge work to work in all of them. So yeah, it's nice. I'll look at them.
Okay, thank you.
Hello. Are you planning to characterize some kind of bioactivity that maybe come from the phenolic compounds that you have in your raw materials? Because I'm doing similar work but with chickpea, and instead of trying to reduce the phenolic compounds, we're trying to quantify them, to characterize them, and also see if they have some sort of bioactivity. That's why I'm asking, because you're considered as an anti-nutrient.
Yeah. So we consider as because it's kind of also an off flavor, because it gives some bitterness that in plant-based dairy alternatives you don't have this flavor of bitterness. So that's also why we try to remove them. But I know that they are considered as bioactive. So sometimes, yeah, the limit between removing something and that sometimes is relevant for some purposes and sometimes it's not relevant depending on the case, you know. But yeah, we are planning to do that. But there's also other projects involved in that, more about biocide stream utilization and yeah, different stuff.
Okay, thank you, and thank you for your presentation.
Thanks.
So other question, or we're done, I think.
Yeah. But thank you a lot. I have been really happy that you were interested in hearing what we are doing at DTU. And I don't know if you know, but we have had the experience of having a tech student in our lab last semester. And we hope for more tech students being interested in coming to DTU and doing, following up on some of the work that Guillermo or Sanne are doing. So if you feel interested to take a semester at DTU, please contact us. We will be happy to have you.
Thank you so much for the invitation. So we hope
Yes.
someone will be there.
Perfect.
So thank you, Sanne and Guillermo. Very, very interesting, your presentation.
Thank you very much for the opportunity.
Thanks.
Yeah, and for
And
the rest, remember that the presentation will be in YouTube if you want to watch it again. Thank you so much.
Thank you. I can see Bastian have joined the meeting. Yeah. Yes. Great. Have a nice rest of the day, and we will probably all DTU people will leave for home. I can see Sanne is home, but yeah. See you guys.
Bye-bye. Have a nice day.
Thank you.
Bye.
Bye.
Thank you.
Bye. Thank you.
Bye.